Hello, my name is Joseph, and I'll be presenting what PCR is on behalf of the Korean HS team. Now, you guys probably have heard of PCR from COVID testing, but there's actually more to that. We're going to first look at what PCR is, next, the procedures behind PCR testing, and the usage of PCR, and of course, the references. To fully understand PCR, we must first ask ourselves, what is PCR? PCR, or polymerase chain reaction, is a technique first discovered in 1983. And as you guys know, PCR is commonly used to detect diseases, including viruses like the COVID. PCR kind of acts like a printer by allowing us to copy a proportion of DNA and make a lot of them. Now, let's take a more in-depth look at how PCR works. The first step is denaturation, where two-stranded DNA separates into two single-stranded DNA at a very high temperature, which is about a 95 degrees Celsius environment. This high temperature melts the hydrogen bond from the nitrogen bases, leading to annealing, which is when the DNA primers combine with the DNA sample from denaturation. At this point, the temperature is lowered to between 45 and 60 degrees Celsius. You might be wondering what DNA primers are, and that's a good question. They define which part of the DNA will be amplified. For example, in terms of printers, DNA primers set which pages to print. The last step is elongation, also known as the extending stage. As the name hints, in this stage, DNA synthesis takes place where DNA primers are elongated. The whole process is done by a machine called a thermocycler to, to rapidly change the temperature for the DNA to be denatured, then synthesized again. Yep, that's the picture of the thermocycler. Finally, we're going to look at the usage of PCR. And it is mainly used for three reasons. First, and most well known usage is to detect viruses in our system, like the COVID. PCR is mostly used to detect viruses that cause disorder related to breathing or respiratory illnesses. This is why when you go to COVID testing, the people that work there insert the stick deep into your noses. Second, PCR is used to copy DNA segments to perform scientific analysis. Without PCR, the DNA segment itself is just too small for scientists to perform analysis. Lastly, PCR also detects disorder other than COVID. PCR is used, for example, to detect genetic disorders. This is possible because PCR testing helps scientists to focus on a specific segments of DNA, which allows them to realize any unexpected genetic makeup that would be unseen otherwise. These were the sources we've used, and thank you for listening. And I hope you have learned more about PCR testing.